Alright, so in this video, we will be discussing the functions of the GI tract, the structures of the digestive system. We'll do a little bit of anatomy review, and then go from the mouth to the stomach, all the way out to the large intestine. So first off, the functions of the digestive system. There's actually a few, maybe generally we think of the digestive part where we're talking about breakdown of food into smaller units, and for us to be able to absorb that food so that we can get the nutrients from it, they have to be in the smallest constituents possible, so like monomers. And so carbohydrates are broken down into the smallest possible bit that can be broken down into, which are simple sugars like glucose. Proteins are broken into amino acids. Fats are broken into fatty acids and glycerol. In addition to this digestion and absorption, which are the major functions, the major goals here, there are some others that go along with this. First off, we have to be able to eliminate food, eliminate the stuff that we cannot digest. We have to be able to move that food through our GI tract. So there is movement here that we'll talk about. So there is muscle action, and we'll talk about those. And then we have to have some chemical component that is going to be, these are kind of tied together because they work together. These chemical components will help with digestion. Also, because this may be time consuming, there is area for storage. So a quick review of the layers of the GI tract. The innermost layer here, we have the mucosa layer, which is shown here. Then, it's this top layer over here. And that is lined with epithelium. But wherever we find epithelium, we're going to find connective tissue with the lamina propria. And the mucosa actually has some muscle in it called the muscularis mucosa. And that's what actually allows these maybe villi to move or to hold their shape. Then deep to that, we have a layer called the submucosa. The submucosa is made out of connective tissue, as you can see in this picture right there. You notice there's some glands in here, blood vessels, nerves, maybe lymph vessels. Superficial to that, on the outside of that, we have the muscularis externa layer. This is an important layer. We have two muscle layers in the GI tract. Generally, there are two muscle layers within the muscularis externa. One is longitudinal, and what that means is there's ones going all along the length of it, like all along parallel to it. And then there's one layer that is circular, and you can think of that as like having rubber bands around the length of your GI tract. And it's important to have these two different muscles to allow for that movement we were just talking about, that movement of the food through GI tract. Last, we have the serosa layer. The serosa layer is a connective tissue layer, and notice that it's kind of connecting the GI tract to the compartment in the body. We're going to go down the length of the GI tract and talk about the important things that are there, and the functionality when it comes to those major functions that we spoke about before. First off, we put food in our mouth, and as soon as you put food in your mouth, digestion is starting. And it has two types, right? There's mechanical digestion in form of mastication, which is chewing, This is a GIF, this little guy chewing. So that's one way, but then a second way is chemical digestion. And our saliva glands actually secrete an enzyme called amylase. Amylase is an enzyme that is going to digest starch into simple sugars, into, I think, disaccharides. I'll just write simple sugars. So that means there's actually a way you can test this, and maybe you'll be doing this in lab. You can put a potato chip in your mouth or a saltine cracker in your mouth, and if you hold it in your mouth long enough while you're salivating, it starts to taste sweet. And what that means is that that starch is being changed to simple sugars via amylase. Our saliva also holds mucus and antimicrobial agents. So then we swallow that food, and swallowing actually has a few stages to it. First off is the oral phase or the buccal phase. Buccal just means cheeks, so it's pretty much just when it's in your mouth. When you do this, it forms a bolus. It forms this like soft mound of food, right? And so it's called a bolus. This phase this phase is voluntary, but the phases that follow it are not. So the phase within the pharynx and the phase within the esophagus are involuntary, and at that point cannot be stopped. And one thing to notice here is that the epiglottis, which is just a tab of hyaline cartilage, is flapping over our trachea and covering up our respiratory tract so that food won't go in there. The act of swallowing is called deglutition. So here it shows it moving down past the epiglottis, and now it's in the esophagus. And the esophagus, if you can remember, is a collapsible tube. And because it has those two layers of muscle within the muscularis externa, peristalsis occurs here. And what peristalsis is kind of like this milking action that brings the food down. It's pushing the food down, 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 down until it reaches the stomach. There is a door here. There is a door or a valve, and that is called the esophageal sphincter. Maybe like right there. And when this door doesn't completely close all the way, this is what can actually cause acid reflux. Your esophagus looks something like this. And if this sphincter does not close all the way, it can make the contents in your stomach rise. It can allow the contents in your stomach to rise up the esophagus. And because what we're going to learn, this is an acidic environment, this can cause a burning sensation because the esophagus is not prepared for this acidic fluid of the stomach. This is what people call heartburn. You know, it's called heartburn because it feels like your heart is burning. And in infants, they spit up a lot because their sphincter is not working as well as an adult sphincter. Just have the stomach. Some important things to note here. First off, so remember we said how there are two layers of muscle within the muscularis externa. The stomach is an exception. It has three. And so there is a third layer here. And that's because a lot of digestion, a lot of mechanical digestion occurs within the stomach, which means the stomach moves. The muscles here contract to allow for, mechanical digestion of our food. And then, of course, there's chemical digestion. In the mucosa, we have gastric rugae, and we're going to take a closer look at this. The stomach is an area where we can store food. If we can, it is where digestion of protein initially begins because it does not begin in the mouth. It can be a protective mechanism because it kills bacteria with its high acidity. It can digest all foods even further because of the high acidity as well. And from here, it changes from a bolus to chyme because it becomes this soupy mixture which is going to go into the intestine. And then there are some interesting factors here. First off, there's going to be something called intrinsic factor, which it will secrete. And something interesting that occurs here is that this is where alcohol and aspirin are absorbed. So as we look closer at our gastric rugae, what we find are these pits, these long pits that occur, and these are called gastric pits. And what we're going to see are there are different types of cells within our gastric pits. So at the top, we have mucus cells, and they're called mucus cells because they secrete mucus. These are goblet cells of, simple columnar epithelium. As we move further down into the pits, we have these specialized cells that are going to produce some special secretions. Parietal cells, they secrete this hydrochloric acid, that acidic environment that we were talking about. They also secrete something called intrinsic factor, and intrinsic factor is important because it helps us absorb vitamin B12 in the intestine. We have chief cells. Chief cells secrete an important inactive enzyme called pepsinogen. We'll talk about why it's inactive in a second. And then there are 3 other types of cells. And ESL cells secrete histamine and serotonin. We'll talk about ESL cells in a second. G cells secrete gastrin, which is a hormone, and D cells secrete somatostatin, which is another hormone. So this is just to show us how hydrochloric acid is secreted. First off, we have carbon dioxide and water within the cell, and via that same enzyme that we've spoken about before, carbonic anhydrase, that is going to change the carbonic acid into bicarbonate and H ions, hydrogen ions. Those hydrogen ions get carried out the apical side via primary active transport, and potassium gets carried in. And so this is active. Requires ATP. And then bicarbonate gets carried into the blood, and this goes via passive diffusion. So this is a co-transporter. This is via secondary active transport. Chloride is going to come in. It's piggybacking on the concentration gradient of bicarbonate. So this is how bicarbonate gets into the blood. So chloride comes in here, and it is piggybacking on the concentration of bicarbonate. And then on this side, on the apical side, it is just going from high to low concentration via facilitated diffusion. And this is how we secrete hydrochloric acid into the stomach. So this is the stomach side. This is the blood vessel side. G cells and ECL cells kind of control the functionality or the activity of parietal cells. G cells are going to release gastrin. That is going to cause ECL cells to secrete histamine, and that will cause the parietal cells to secrete hydrochloric acid. This is going to reduce the pH of the environment within the stomach, which is allowing the proteins to be denatured. Remember that chief cells are going to release pepsinogen, and pepsinogen is in its inactive form. And it is inactive, it is released in an inactive form because it is going to digest protein. That's its job, it's digest protein. And you wouldn't want it to be active when it's inside the cell, or else it can, degrade the inside of the cell. And so it turns into pepsin, it converts into pepsin, and it converts into pepsin when it comes into contact at HCl. When it comes into contact with HCl. So pepsinogen is converted into pepsin with the introduction of hydrochloric acid. And then when we have even lower pHs, pepsin is even more active. So here it shows the mucosa layer, the chief cells within the mucosa layer secreting pepsinogen, the parietal cells secreting hydrochloric acid, and that is going to convert pepsinogen into pepsin, which is just going to break down protein. And this occurs with the introduction of hydrochloric acid because we wouldn't want an active protein digester within the cell. So why doesn't the stomach eat itself? First off, we talked about how pepsinogen is inactive inside the cell. But why doesn't the hydrochloric acid eat up the epithelium of the mucosa of the stomach? Well, we spoke about how there are mucus cells at the surface of those gastric pits. The mucus coating is going to protect the epithelium of the stomach. It contains, first off, bicarbonate that is going to neutralize any hydrochloric acid nearby. And it is a barrier so that pepsin cannot degrade those epithelial cells. The tight junctions, there are tight junctions between epithelial cells. So I can draw, epithelial cells here that prevent hydrochloric acid and pepsin leakage. So it's really, and this is mucus maybe, it's really controlled where hydrochloric acid is allowed to go within this area. And these cells, these are epithelial cells, so they are able to do mitosis a lot, and they replace themselves about every 3 days. And maybe you've heard of intermittent fasting, but one of the reasons behind intermittent fasting is for this renewal of your digestive epithelium. So perhaps you've heard of stomach ulcers before, or peptic ulcers. And what they are when the mucus does not protect the epithelium of the stomach. And what that can do is their hydrochloric acid can impact those areas. And so you can treat with a bicarbonate like Tums. Tums is bicarbonate, sodium bicarbonate. And so that is the job. The job there is to buffer the acid. Tagamet and Zantac are going to block the histamine of those ECL cells to reduce HCl release. And Prilosec and Prevacid inhibit the potassium hydrogen pump, so it also reduces the the acidity within the stomach. And antibiotics are there to treat bacteria. In the small intestine, this is where absorption is going to occur. So we've been talking about digestion in the mouth, digestion in the stomach, but here is where absorption is finally can occur. So for absorption to occur, the small intestine is equipped with an increased surface area. And so I always like to say it's kind of like a scrunchie. You can stretch it out, and I believe you can stretch out the small intestine to like the length of a tennis court. These are really folded in on themselves. These are called the plicae circularis, or the circular folds. And then when you look at them beneath the microscope, they're even more folded on themselves with these villi. These villi even have microvilli, and in the microvilli, they have this border where they have digestive enzymes. And so the stomach is digesting with the chemical digestion from all those constituents, as well as the mechanical digestion, because remember there's a 3 muscularis external muscle layer here rather than the typical 2 muscle layer. And so there's a lot of mechanical and chemical digestion here, but still it's not completely digested to the absolute smallest constituents. And that is going to occur here in the small intestine using the digestive enzymes of the brush border of the microvilli, as well as, and this is going to be in the next video, the digestive enzymes of our accessory organs. So our pancreas, our liver is going to help out as well. And gallbladder, actually. So here we have a close-up of one villi. You can see the simple columnar epithelium here with goblet cells. Goblet cells are still present within the duodenum because there is still acidic components coming off of the stomach. And then remember that we are going to want to break down our constituents into their smallest monomers, and we'll talk more about those when we talk about the accessory structures. But one thing to notice here is that there is a lymph vessel called a lacteal surrounded by a capillary network. And this is shown because fats are not small enough to travel within our capillary network. And so fats actually travel within lymph. They travel through the lymph and they come in via these lacteals within our small intestine. Individuals who are lactose intolerant are lacking lactase enzyme within their brush border enzymes. And so what happens is lactose goes undigested and it goes into the large intestine. And this can cause diarrhea, gas, and cramps. And this causes diarrhea, gas, and cramps, or, well, let's say it causes gas, because what happens is the bacteria in the gut can have lactase, and so they will digest that enzyme, they will digest the sugar, and then, gas is a byproduct of that. Diarrhea occurs because the presence of this sugar is going to draw water to it, which causes diarrhea. All right, so we've spoken about peristalsis, which is kind of like a milking action through the GI tract, and this begins in the esophagus and continues on in the small and large intestine. But there is a second type of movement that occurs within the GI tract, and that's called segmentation. Segmentation, I like to say, say for example, if you had a roll of cookie dough and all the chocolate chips were on one end of the cookie dough, you would squeeze it right here to try and move it down, right? And then squeeze it back and forth, and maybe on the ends and then in the middle, and then on the ends and then in the middle, on the ends and the middle, trying to distribute the chocolate chips evenly throughout your roll of cookie dough. This is kind of what the action and the purpose of segmentation is. The goal here is to mix the chyme and the mucus with the digestive enzymes within the small intestine. Because remember, things are not completely digested yet. There's still digestion occurring. And so the goal is not to move things down the line, down the way necessarily. That's the role of peristalsis. The goal here is just to mix things together. And so, movement is relatively slow, and the action of this is actually caused by these cells in the muscle layer called the interstitial cells of Cajal. The interstitial cells of Cajal. And these cells are kind of like the cells of the SA node, where they're kind of these pacemaker cells, where they're always sending an action potential every, however long pace, every 5 seconds. And so similar to the SA node, there is like a beat of a drum that the interstitial cells of Cajal kind of fire off, which allows for this constant, consistent action of segmentation in the small intestine. This is just to show you what the action, the slow waves of the interstitial cells of Cajal do. And notice how long these are. These are not in milliseconds like we've been discussing in other parts of the body. These 3 waves take 5 seconds, where you could actually, see these in action with the naked eye. So these are setting a pace. They do set a pace, but they set a very slow pace. This is what the interstitial cells of Cajal look like. They are setting that slow wave action, and, like 5 seconds for 3 waves. And it is going to cause depolarization of the smooth muscle cells here. And then we also have an autonomic axon with its varicosities. If you can remember what the varicosities are, it allows for action potentials to occur at all of these, so that there's kind of like action occurring for all these muscle cells at the same time. And so absorption of nutrients occur in the small intestine, and mainly when, the chyme gets to the large intestine, this is pretty much waste. The only things that are absorbed here may be electrolytes and water. Water is still absorbed here. There is active transport of sodium, and water is going to follow that, just via basic osmosis. So there is no digestive function here. By the time food, you wouldn't even call it food now. By the time chyme is in the large intestine, this is now considered waste. In the large intestine, this is where we have our largest population of microflora, biota, bacteria, where you can have over 400 different species of bacteria. Some individuals have less. This is why people might take a probiotic for gut health, and we'll talk about that in just a second. This actually just happened to a friend of mine a week ago, where she had an appendicitis. So I should say that the appendix, I'll show you, the appendix, which is actually called the vermiform appendix. Vermiform just means worm-like because it looks like a little worm. It's literally like smaller than the size of your pinky. The vermiform appendix, we don't really know what the function of it is. It's this structure that, evolutionarily has been getting smaller and smaller in humans. We assume or we think that it used to be a holder of good bacteria, a carrier of good bacteria. And so, some individuals have infection of that appendix where maybe it gets infected or inflamed. And this, can be detected because it just gives sharp pain in your lower abdomen. If your appendix ruptures, that can release bacteria because it does contain bacteria in it. And so that can effectively cause sepsis. It can be really dangerous. Or it can cause peritonitis, which is the inflammation of the peritoneum, which is the connective tissue layer surrounding our GI tract. I think this is a really important thing to talk about because people assume that antibiotics are good no matter what, but it's actually not true. Antibiotics should only be used when you actually have a bacterial infection, like a UTI maybe, because antibiotics are going to not only kill good bacteria along with the bad bacteria, but they can also select for bacteria that will withstand the presence of antibiotics. And so, maybe you've been reading in the news like the rise of a superbug or bacteria that we cannot kill with antibiotics anymore. And that's because there has been an overuse of antibiotics in our country, in the world. So when you are sick or when you do take antibiotics, make sure it's under the supervision of a doctor, prescribed by a doctor. And also, it's always like for 5 days or for 7 days or for 10 days. Make sure you take your full amount of antibiotics. Take all the pills when you're supposed to do it. Because if you only take 5, like for 5 days instead of all 7 days, you have not killed all the bacteria. And you may have selected for the bacteria, you're allowing for the bacteria that were the hardest to kill to survive and then to replicate, to undergo mitosis and to divide. And so it's really important to take all of your antibiotics when you prescribe them. Only take it when needed, and when needed, take them all. Microbiota are important. And again, I just brought up the use of probiotics. But they can produce for us some vitamins like vitamin K. They can produce folic acid. And so they are beneficial to us too. We live in a symbiotic relationship with the bacteria, with some of the bacteria in our gut, most of the bacteria in our gut. They can ferment indigestible food. And by having the presence of good bacteria in your gut, you reduce the capability of bad bacteria to be in your gut. So it's a good thing to have good bacteria, again, which is why people purchase probiotics and use those as a dietary supplement. They can have anti-inflammatory effect. They can help intestinal epithelium. And I will share that there's also research about intestinal gut health and brain function. And so they can possibly impact how our brain functions. Last, we have defecation, and this is going to be caused by a mounting pressure due to that buildup of waste material within the rectum, and that'll allow for your anal sphincters to relax. Your inner anal sphincter is involuntary, while your external anal sphincter is voluntary, of course, thankfully. And then contraction of pelvic floor muscles, rectal muscles, and abdominal muscles like your rectus abdominis or transverse abdominis 2 are going to raise the pressure and allow for defecation.